Hey, my name is Tyler Garwood, and I am a PhD candidate advised by Dr. Tiffany Wolf. Moose are culturally, ecologically, and economically important to Minnesota, but their persistence in the state is threatened. Recent studies undertaken independently by the Grand Portage Band of Chippewa and the Minnesota DNR both found that Paralophus strongylosis tenuis infection is the leading cause of death among adult moose in Minnesota. P. tenuis is a parasitic nematode that reproduces asymptomatically in white-tailed deer and requires a slug or snail intermediate host to complete its life cycle. But when a moose accidentally consumes an infected slug or snail, it can suffer neurological damage and death. Infections in moose are becoming more common due to increased overlap in deer and moose habitat due to climate change and land conversion. Management options to reduce P. tenuis transmission to moose are currently limited to reducing deer densities, which is often politically infeasible and varies in efficacy. New intervention options are needed. Habitat management to reduce transmission is less controversial, and previous research found that increased moose use of certain forest types predicts P. tenuis infection probabilities. However, whether those forest types or other landscape features serve as barriers to or corridors for P. tenuis transmission remains unexplored. To understand how landscape features affect transmission, we collected a spatially stratified set of P. tenuis larvae from across the Grand Portage Indian Reservation. We sequenced DNA from those samples and identified locations on the P. tenuis genome that reliably vary between individuals. These genetic variations allow us to infer gene flow between P. tenuis sample locations. Gene flow results from P. tenuis movement, survival, and reproduction, and therefore is a good measure through which to study transmission. When patterns of P. tenuis gene flow are mapped on the landscape, we can begin to develop hypotheses of which landscape features shape those genetic patterns. For instance, we might hypothesize that steep topography reduces gene flow if samples collected on either side of a steep ridge exhibit little gene flow. After formulating several hypotheses about how the landscape shapes P. tenuis gene flow, we test the relative merit of these hypotheses through a model selection framework. To that end, we collected 139 P. tenuis larval samples. We then identified about 110,000 genetically variant sites, which we used to delineate 5 genetically distinct clusters of P. tenuis on the reservation. 4 of those clusters seem to be concentrated on the outside edges of the reservation and near clear cuts and rivers. However, we still need to assess the effect of different landscape features on P. tenuis transmission through our modeling framework. Our results can be used to predict and enhance the efficacy of habitat management plans aimed at reducing P. tenuis transmission to moose. If we find landscape features that shape brainworm transmission and are manipulatable, we can use that information to manage the landscape to reduce P. tenuis transmission. For example, if a forestry treatment is associated with decreased P. tenuis gene flow, it may be beneficial to moose to increase the presence of that treatment on the landscape. And even if the features are not manipulatable, we still gain valuable insight on where transmission risk is high or low. This study is particularly relevant to the 1854 ceded territory, an area covering most of northeastern Minnesota, where the Grand Portage Band, important partners in this research, and other Chippewa tribes retain treaty reserved rights to preserve their culture and subsist through moose hunting and other forms of hunting, fishing, and gathering. Thanks for listening, and feel free to reach out with any questions or thoughts.